Hey guys! We're going to be starting with the Dalia Science Neuro short soon. So I've got a wee presentation here, and in the beginning I'll just try to say a few things about the whole series, why it came about, and why I'm going to be doing it, I think. So first of all, the whole idea for the series came because I shared on my stories that I'll be doing tutorials this Trinity term. So Trinity term is the third term in the Oxford calendar. And yeah, I was told that there's this exchange student who was supposed to come to Oxford, but they won't be able to do it, obviously because of the virus situation. And so yeah, so and they asked if I would like to do tutorials with that student in neuroscience, and I said yes, obviously, as you know. And so yeah, just to let you know a little bit about Oxford tutorials in the first place. So Oxford tutorials usually last for maybe an hour and a bit. And during this tutorial, students first of all send a piece of work to the tutor before the tutorial, like an essay or like a set of filled in questions. And then the tutor would check this homework, check this essay or whatever it is, and then they would send it back to the student before the tutorial, and the student will be able to look at their work, look at how their work went, like what they did well, what they did wrong. And then in the tutorial, when you turn up to meet your tutor, usually it would be 1-to-1 or 2-to-1 or 3-to-1, so a small group meeting. During the tutorial, you would discuss that essay, whatever it is. And so yeah, obviously now that we're all online, these tutorials have a bit of a different form. So yeah, so I just basically talk to the student online and we discuss a lot of concepts. And so this is what I'll be sharing here in this Neuro short. And yeah, so just as a disclaimer, this is not really going to be exactly like an Oxford tutorial, but it will be something like that. And yeah, and I'll be able to share some concepts that I would discuss with the student with you, and I will be able to present some stuff to you. Obviously it will be much shorter than an hour. But I want to just share a little bit of an introduction to the topics that I want to discuss with you. And then after the tutorial, after this little Neuro short, I'll be able to send you some papers and some stuff that I'm discussing here for you to look at it later and check it out. I see there are some people from my high school, hello. And yeah, regarding the questions, I'd like to say that the talk, which I'm going to give, will have like 3 parts. And I won't be exactly able to look at all the questions as I go because it's a bit distracting and I want to share some information with you. But I'll be looking at the questions in like increments. So the talk will have 3 parts, as I said, and I will look at the questions after every part, and then I'll be able to answer some of the questions. So I think this is a good moment to start. So welcome to the Dalia Science Neuro short, and today we will be doing a bit of an introduction to neuroscience and to fruit flies, because fruit flies are what I work with. They're my model organism. And just to start off the bat, what is a model organism? A model organism is an organism that you use to model different biological phenomena on. So obviously it's wrong, immoral and unethical to work on human beings, despite the fact that sometimes some scientists work with some human cells, but that's got nothing to do with a whole human being. You wouldn't be able to work on a human being. But what we are interested in fact, are human beings. We want to find out about phenomena related to humans. And so in order to do that, we are interested in model organisms. And model organisms are organisms which you can look at, you can work with them rather than a human, rather than something else, and be able to understand some phenomenon that you are curious about, such as, for example, some stuff related to neuroscience. So examples of model organisms could be flies, they could be mice, they could be ferrets, they could be tadpoles and frogs, and there are other model organisms like yeast, which people work on. So, first of all, first off the bat, what is neuroscience? So I said we're going to talk about a bit of neuroscience, we're going to talk about fruit flies. Well, neuroscience in general is the biological study of the nervous system, and it can also be called neurobiology. But in fairness, this is such a broad definition because it could mean a lot of things. It could mean people working on brains, but it could mean also people working on individual nerves. It could mean people who are in clinic and they work with patients, and the patients go into a mRNA machine, not mRNA machine, MRI machine, what am I talking about? So they go into an MRI machine and they get a scan of their brain, and that's also neuroscience. But I don't work on neuroscience like that. I work on neuroscience, which we could call more molecular neuroscience. So I work with with individual neurons and individual cells that I look at in flies, in the fly model. So yeah, the model, someone's asking what's the model that I'm using in my project, and that's fruit flies. So yeah, neuroscience is a very broad definition and it could involve a lot of things. And so I today will explain a little bit about the molecular neuroscience that I work on. So let's start with some basics about neuroscience. Well, I am hoping that most of you would know maybe that the very basic cell in the nervous system of the human body and the bodies of all the organisms that have a nervous system is a neuron. And the neuron is an excitable cell. It's a cell that can conduct electricity, it can conduct impulses, and those impulses are basically signals which go from the cell body towards the end of the cell, and they carry some sort of an information. So for example, my hand moving right now, that's because my brain is sending impulses to my hand and letting it move. And so a neuron would have different elements which we can quickly discuss and look at. A neuron would have the soma, which is the cell body, which is here. And the soma contains the nucleus, which is where the DNA is, obviously. And outside of it, there are dendrites, and they are the little connections which allow one neuron to connect to the other and convey information from one neuron to the other. And then further, we've got the axon, which is the extension of the neuron. And this is the little nerve, the line along which the impulses are being sent. And in an axon in vertebrates, so organisms like me and you, humans and so on, you would have this myelin sheath, which allows the impulses to travel faster along the nerve cell. And then finally we have the Schwann cells, and they are a kind of a glial cell. And a glial cell is a supportive cell for the neuron. It takes care about the neuron. Sometimes it takes care of the debris clearance, which means that it cleans up the neuron if there is some sort of dirt around it, we could say, and also supplies nutrients to the neurons, as well as forms that myelin sheath, which allows those impulses to travel faster. And this myelin sheath, I don't know if it's visible, it's created in little kind of nodes, and you can see that there are breaks between the pieces of that myelin sheath, and they're called the nodes of Ranvier. And that's very interesting because thanks to those nodes of Ranvier, the impulses, the electrical impulses, can travel much faster along the neuron, and that's because they skip from one of those pieces of myelin to the other, and they go much faster. And then finally here we've got the axon terminals, and if we imagine that there's another neural cell here and another neuron here, these axon terminals would connect to the dendrites of the next neuron. So this is basically what a basic building block of a nervous system is, and this is what we'll be working with, and this is what I work with when I look at my flies. And so there are two very important elements and divisions in the nervous system, both in the human one and in the fly one, and in any actually system that you could look at, in any organism that has a properly formed nervous system. And so the first element which we would like to look at of the nervous system is the central nervous system. And the central nervous system in the human beings would be comprised of the brain, so that's here, the brain, and the spinal cord, which is just this tube which goes along our spine, basically. And so this is the central nervous system, and that is sort of the part of the nervous system that's responsible for the production of those signals. So that's where the signals originate from our brain. As we know, the brain is responsible for thinking, but also the signals, as I said, that are controlling my hand, my movement, my taste, me smelling something, they come from the brain, and they go outside to the organs of the body, to my hands, to all the other elements of my body. And you can see that on this plan here, we have a clear comparison where the human central nervous system is quite comparable to the fly central nervous system, even though fly is quite distant if we think about the animal kingdom from human beings. So therefore, despite its simplicity, we can compare the human nervous system to the fly nervous system and do what I mentioned before, which is model certain things on it. Diseases, model the development of it, and so on and so forth. And then there's another element of the fly nervous system, and that is the peripheral nervous system. Sorry, my slides got kind of twisted around here. Let me come back. There we go. And so the peripheral nervous system is the second part of that nervous system that I told you about. So we've got the brain, we've got the spinal cord, and we've got the fly equivalents to those. And then we've got the peripheral nervous system, and this is the element of their nervous system which is responsible for, first of all, making things do stuff, such as making my hands move, making my skin feel cold or hot or other things. But it's also responsible for sensory information. So that is, if I touch something cold, the peripheral nervous system is responsible for my hand the impulse from my hand being sent back to my brain to tell my brain about the fact that I'm touching something cold. So these are the two main elements of the nervous system. This is what we're starting with. And yeah, and so these are the building blocks which we could look at. So this is the first part of my talk, the building of the build up, the sort of the structure of the nervous system. So I'm going to quickly see if we've got any questions here that people have about the nervous system, the neurons, all kinds of things. I don't see that many questions. People are saying it's interesting. That's really good. I'm trying to see, yeah, which model I use in my project. That's been asked by user at Dalia. Yeah, anyway, so I said I'm using fruit flies. This is what I work with. I work with fruit flies. And then someone's coming to Oxford in September, the next press. Congratulations, and I hope to see you soon. And I'm seeing, one second. And I'm seeing here, oh, someone says it's a great explanation. Thank you very much. Anyway, you can keep the questions coming. And if I don't see them now, I'll try to see them as we go. Someone has a question about glial cells. Please feel free to write it down, and in a minute I'll be able to probably answer it. Actually, glial cells are what I work on, and they're my specialty in my project. So anyway, now we're going to move to the fruit flies. So yeah, glial cells. I've got a user, it's Shamsul, asking me about glial cells. So anyway, yeah, the fly nervous system is similar to human nervous system, as we've seen in the slide before. There is a comparison between those two, and it's similar enough, in fact, that we can use flies to model a lot of nervous system related diseases, nervous system related things, including the development and including some diseases. And we will be looking at that actually in further episodes of my Neuro short. So anyway, let's go back. No, let's go forward. So now we're moving to the part of the talk where I'll tell you a bit about the model, which I mentioned before, so about the fruit flies. So I work with fruit flies, they are my model organism, and there are many reasons why people work with them. And first of those reasons is what you can see here in this slide. So the important thing about fruit flies is that you can easily distinguish the males from the females. And we can see here that we've got a female here and a male here. And basically, we can see that there is a difference in how they look, and that is easy to distinguish on a scope in the lab. You can go and see and look at the flies, and you'll be able to distinguish between the two. And that's important to know, because when you are working with flies, and let's say you would like to model some disease or some process on flies, you might want to perform specific crosses. And those crosses will just mean that you will cross two types of flies of different genetics that you are looking at, that you're interested in. And so then it is very important to be able to distinguish the males from the females, so that you know which genes you're crossing in between the two, which genotype you will get in the end. So that's one of the very big advantages of working with flies. Furthermore, there are other important things regarding the research on flies, and here's a little bit of a fly history. And so this research on flies basically started with Thomas Hunt Morgan, who in the beginning of the 20th century, so I'm saying here that in 1910, he started looking at heredity in flies at Columbia University in the United States in New York City. And so this research of his, as well as other research, led to as many as 8 Nobel Prizes awarded for the research on fruit flies, which I think is quite a lot, considering how simple of an organism fruit flies are. And here in this colorful image, you can see the distribution and the patterning in an embryo of a fly. And that's one of the earliest experiments which were awarded with the Nobel Prize. Actually, it was actually not the earliest, because it was in 1995. So there's a group of researchers, some of whom were from Switzerland, who looked at the patterning of an embryo in fruit flies. And so thanks to that, they understood some of the very important principles of development, not only of flies, but also of other organisms. So I would say that for how small the fruit flies are, and for how simple they are, you can do a lot of very important and interesting research on them. Someone's asking if I can share their slides, and I think I can, but I might need to edit them depending on what info I've got in them. Anyway, let's see what we've got next. Why would we want to work with fruit flies? There are a lot of advantages to working with fruit flies. And so first of all, they are easy and cheap to work with. They're easy to work with because despite the fact that they can fly away, you can easily anesthetize them. What that means is you can put them to sleep while you work on them. So when I go to the lab, when I work on fruit flies, I can put the flies to sleep and I can look at them, I can decide which ones are females and which ones are males. And there's another important thing, as I said, about the flies. So the males and the females, first of all, are easy to distinguish, but also there are some features of their bodies which we can relate directly to their genetics. So we can also look at their, for example, wings, we can look at the appearance of their head, their eye, we can look at their appearance overall and the color that they are. And so these are markers which are linked to the genetics. And so I mentioned that the flies have very powerful genetics, and that is true. Indeed, working with flies allows us to perform a lot of genetics, which would be very hard to do or very time consuming if you worked with mice or if you worked with another organism. So yeah, another advantage to working with flies is the fact that they reproduce really fast and they have quick development. So the generation time, which means the time from when you get an embryo fly until that fly is an adult, only takes 10 days. That's very fast. And the flies mature really quickly and they are able to produce a lot of offspring. And that's important if you're doing screens or if you want to test something more than one time. Recently I had a post where I explained why research takes a long time. And that is one of those points that I made, was that research takes a long time because you need a lot of repetitions. And so flies are really helpful in that, because if you use flies, you can have so many of them, you can have them reproduce really quickly and develop a lot. And therefore you can see a lot of the same of the phenomena that you want to look at in many flies. It's harder to do with, for example, patients. If you wanted to take certain images of a brain from 10 patients, it might be hard to get that many patients. Sometimes some people struggle to get some patients at all, which is quite problematic. So anyway, yeah, this is why it's important to have fast development, quick development, and this is what flies are really good for. And then I mentioned the genetic markers, which facilitate experiments, and then those markers are clearly visible as phenotype. And phenotype is a very important kind of expression in genetics. And phenotype just basically means the appearance. And so, for example, my phenotype is that I've got brown eyes, brown hair. And in flies, the phenotypes have got to do with, for example, how their wings look like, what their eyes look like, and so on and so forth. And so you can relate the genetics that you're interested in directly to the appearance of the fly, and that's a huge advantage. And finally, many useful genetic tools for fly research already exist, and that's an important thing, again, because rather than having to develop those techniques and develop those tools yourself in the lab, which takes time, energy, and it costs a lot of money, you can simply go online and order certain fly lines, order certain reagents which can help you work with the flies. And so that's really important because it saves you time and it makes your research much easier for you. So anyway, and then there's something very important which I'd like to mention, because a lot of people say, well, humans and flies aren't that similar to each other. Well, that's true to an extent. But what is also true is that 75% of non-human disease genes have a recognizable match in the genome of fruit flies. That doesn't mean that we, us, humans, and the fruit flies are 75% identical. It just means that some elements of the genome of the flies are similar enough to the human genome that we can go and work on them and make conclusions and find information which will relate back to the human beings, and it will help us with drug development, with understanding the development, with understanding diseases, with understanding mutations and other things. So this is a huge advantage of flies, because for that reason we can use them in a broad variety of all kinds of research relating to flies. Anyway, so that's the second part of my talk regarding why fruit flies are a good model for working on in terms of disease, in terms of biology. I'm going to quickly see if I've got any questions that people might have. There isn't any discussion about the COVID-19 because I'm not a virologist and I won't be talking about that because I'm not an expert on it. I talk about stuff that I know and that I'm comfortable with and that I can answer questions about, so sorry about that. That was a question from, it's Hamsul, sorry. Some people are saying that they work with mice brains, and yeah, so some of the most important and interesting experiments which you might have in science, in biology, in neuroscience, is to compare if, for example, what you saw on flies relates back to what you can see on mice, because mice obviously are vertebrate organisms, while flies are insects, so they're a little bit further away from humans. But then if you find something in flies and it still is true and it still rings true and it still looks the same in mice, that means that there's a huge chance that it's conserved, which means it's preserved between different groups of organisms because it's quite a basic function. And therefore, we might think that it would be the same in human beings. I already said that I'm going to try to share the PowerPoints, and someone's asking me, neurons in flies must be microscopic. That's the Hustler grinder, but a very funny username, by the way. And so yeah, they are quite small. They're smaller than the human neurons, obviously, than the mice neurons, but it turns out that their arrangements and how they look and how they interact with each other are quite similar often. So despite the fact that they're smaller, they might produce similar structures and similar circuits in the brains. Okay, so a lot of people face neuron weakness. What's the main reason for it? I'm sorry, I don't exactly know what you mean by that. If you could specify your question, if you could rephrase it, maybe I'd be able to answer it. So we're going to move to the third part of my talk, which is why are flies being used for neuroscience? And there are, again, many good reasons for that. And one of them is that the flies display a rich set of behaviors. And we could think about flies as quite simple because they're so small and they don't appear very complex to us when we, at the first glance, I would say. But I would say, and the research is being done on flies, which concerns quite complex behaviors, or at least the appearance and the sort of formation of quite complex behaviors, the very baseline of those behaviors. So we'll look at some of the behaviors that the flies have in a minute, but yeah, flies have a rich set of behaviors, in fairness. And neurons and circuits are genetically identifiable in flies. So what I mean by that is the fact that you can go and find particular individual cells which belong to a particular circuit that you're interested in using the powerful genetics that I mentioned before. And so that means that you can go and order lines of flies that you're interested in and cross them together and then be able to go and find a specific neuron somewhere there in the fly brain that's responsible, for example, for smelling. I was listening to a talk which I actually shared in my stories recently from the Oxford Cortex Club, where they went and looked at a specific structure in the fly brain which is responsible for active sleep and deep sleep and different elements of sleep. So that's quite cool, and therefore there's this powerful element to this genetics of the flies that you can use to your benefit as a researcher. Finally, there are transgenes which are available to help dissect functions of circuits. And that means that using a transgene like that, you might be able to find out, for example, what happens if I eliminate that one cell that I was interested in. So you can genetically encode, for example, elements and substances which will kill that particular cell, and you can see what will happen. You can genetically encode fluorescent proteins which will enable you to visualize that particular cell that you're interested in. And yeah, so this is a huge power of the Drosophila genetics. Again, by the way, Drosophila is fruit flies. I hope that I explained that. Probably not. Regardless, Drosophila, that's fruit flies. So yeah, so having transgenes, having this genetics makes Drosophila be on your side. This is a really important benefit which you can get from working with the flies. And finally, you can measure physiological activity of neurons in behaving live animals. So you can literally have a live fly, and then you can have it on a stage, and then you can put electrophysiology equipment in its brain or in its muscle, and you can measure what those neurons are doing. And that's very important as well, because it might be much harder to do things like this with mice. And even if it is doable, it might be more expensive. So flies are like a really great platform to look at behaving live animals and make connections between their behavior and the activity of the neurons to the smallest neuron available out there. And so now we're going to quickly look at some behaviors that the flies have. So courtship, that's one behavior that the flies have, connected to mating. Is that mating? This is mating. So courtship and mating come together, and they've got to do with the males, for example, moving their wings in a certain way, which creates the song that the females recognize, and that has got to do with them mating, them getting ready to have sexual behavior. And basically that's really interesting because the flies are quite simple. So obviously there is this circuit somewhere there in the brain, some behavior of the brain, which was responsible for that courtship, which was responsible for why the female decides to, for example, mate with that particular male. Another thing which we've got is dominance and aggression. So we can see that flies often display dominance. For example, there would be like a dominant male who tries to sort out its territory, try to appear like the strongest one of the pack. And then that's got to do with the aggressive behavior and with fighting. And this is very interesting as well, because there's plenty of research which looks at this dominance and aggression in terms of the circuits and in the context of the circuits which also exist in the human brain, to try to understand why aggression arises in the human brain, what gives rise to the human aggressive behaviors. And if we can figure it out on such a small level as a fly, as some kind of a little insect, well, it must be a very basic, very conserved circuit then, and we can relate it back to humans or relate it to mice. Finally, they also appear to have some reward-related behavior, and that's been shown in experiments where, for example, they were given a little bit of sugar, and they've shown a positive reward behavior. So they would go and like they would appear in the same place where the sugar was given to them before. And there was also experiments where people shocked them, like gave them a little electric shock, which is a bit awful, but you know. As I always repeat, people do these on flies and not on humans because of the fact that this is a bit controversial, and yeah. Anyway, so there was experiments where people did a little bit of a electric buzz to the flies, and the flies were avoiding this buzz, avoiding the place where they got that buzz. And furthermore, we can see here that there is this graph which basically shows a mammalian olfactory bulb, which is labeled A, and I think that's this one. And then it also shows the insect antennal lobe. And as you can see, so this is a mammalian, so this is from a mouse or from a human being. This is showing how this olfactory bulb looks like. Olfactory bulb is the element of the brain which allows the animal to smell things. And you can see that there is striking, significant similarity between the insect olfactory system and the mammalian olfactory system. So you can see that, again, even in terms of the structure and the outline of those structures in the brain, there is a lot of comparison between the mammalian systems and between the insects. Anyway, and finally, we can see some more important things which you can model on flies, and they are circadian rhythms. So for example, in my lab where we keep the flies, there is an automated light system which turns on at 8 a.m. and turns off at 8 p.m. So that's in place because it mimics the daytime and the nighttime for flies, because otherwise, what's interesting, they might have disrupted development and behavior. So flies need to see the daylight and the nighttime in order to develop properly. So you can model that and you can disrupt it in order to be able to understand what regulates those day-night rhythms and day-night behavior in flies. Another thing which you can model, which is what I work on and which I'll be talking about next time in the talk, is the learning and memory, and that has got to do with a phenomenon which is called synaptic plasticity. So I work on synaptic plasticity, and synaptic plasticity basically has to do with the formation and the destruction of synapses or connections between neurons. And therefore, using flies, you can go down and understand the basics of processes such as formation of new connections between neurons, which relate directly to learning and memory, and I think that's really quite cool. And yeah, behavior, connecting to the smallest cells and individual proteins being labeled in a fly cell on a fly system. This is one of the biggest advantages in terms of working with Drosophila, because you can go and look at these individual neurons and directly compare them to some behavior or some effect which you see in terms of the whole organism. And then finally, we can look at sleep, and I've been sharing this talk recently where the researchers looked at the fly sleep, and you can model sleep in flies to the extent of even understanding the deep sleep and the active sleep, and see what changes appear in the fly brain as the fly falls asleep and then wakes up and then falls asleep, and what it looks like on a neuronal level down to a single cell again. And finally, there were studies which looked at drug and substance abuse with flies. So for example, flies which were exposed to alcohol or other things, which can help us model those behaviors in humans, because drugs and substance abuse obviously is a complicated behavioral as well as neuroscientific issue with human beings. And you can use flies to try to model it and try to understand it better. So anyway, this is all I've got for today. This is the part 3 of my talk, which explained why flies might be good for neuroscientific research. And if you've got any questions, you can ask me now. And thank you so much for tuning in. A lot of people actually tuned in. I really appreciate it. Thank you so much. And yeah, I will be carrying on with the lives probably on Saturdays. I think it's a good time. I think we can be quite a good time to have a live. And next time we will be looking at some specific behaviors, which I mentioned, and specific things which you can go and model in fruit flies. And we might look at the learning and memory or sleep. That's what I've been thinking about. So let me know if you've got any particular behavior that you're interested in that you would like to hear about in terms of it being modeled in flies. Yeah, someone says that Adel Ahmed 94 says, I hope you have a channel on YouTube to upload this video. I'm so interested. Yeah, I was thinking about it actually. I'll go and listen to it after it's done just to see how well I've done. And if it's okay, I might upload it to YouTube. And then yeah, Bine user says that behavioral studies are really important in research, and that is very true. And that's what I said about flies, that this is like a really good model for that, Drosophila fruit flies. Anyway, I am seeing, are there any more questions here? Someone's asking what about the fish nerve system. Yeah, there are neuroscientific studies done in zebrafish. I'm not an expert on those, but I know that one of the advantages of zebrafish actually is the fact that you can have them they are transparent. You can have them genetically done such that they are transparent, and therefore you can do whole animal imaging, which is quite interesting, because if you have like a fluorescent protein in zebrafish, you'll be able to put the whole thing pretty much on the microscope and look at it and look at the nervous system labeled fluorescently. Yeah, someone says that there's drug and alcohol abuse, and that they're interested in it. I can go and look up some studies and post them on my story if you'd like to read more about it. And do you ever work with Drosophila larvae or only adult flies? I only work with larvae, actually. I never work with adult flies, but I'm also interested in the adult flies because I think there is an entire range of things which you can't do with the larvae that you can do with the adults. And so, for example, you couldn't really do any behavioral studies or anything like that on the larvae because they're just too simple. And so I use larvae for studies of synaptic plasticity. They're quite a famous model for that. But a lot of people do all kinds of crazy and interesting behavior things on the adults. How many years to finish a PhD at Oxford? Usually it's from 3 to 4 years. My PhD is supposed to be 3 years. How can we detect if there is any disease in model brain? Well, usually if you would like to look at a disease, you would genetically modify your model to have the disease. So in flies, you can, for example, order specific fly lines which have modifications where they were given, for example, a protein which mimics Alzheimer's disease or Parkinson's disease. You can look at models of ALS, which is the motor neuron disease. So that's the disease that Stephen Hawking had, which basically leads to the disappearance of people's muscles and they are unable to walk or anything. You remember he was in a special kind of a wheelchair, which was built by Windows for him. And then I'm trying to see how many years. Please have a session about admission in Oxford. Yeah, okay, I might consider that. It obviously wouldn't be in the Dalia Science Neuro shorts because they're about neuro. So anyway, thank you so much everyone for tuning in, for listening to my talk. I hope you liked it. Please feel free to send me feedback. Feel free to let me know if you enjoyed it and what you would like to hear about for the next time. And yeah, I'll be posting some papers and some stuff that I was talking about in my stories soon. And thank you so much, everybody, for tuning in. Bye bye.